Erythrocytes, or red blood cells, are the major portion of formed elements found in blood. That is about 95%. Charged with delivering oxygen to cells and removing carbon dioxide, red blood cells, or erythrocytes, are critical to survival. Its importance can be emphasized from the fact that a single droplet of blood contains about approximately 5 million erythrocytes. Let's look at its shape. The diameter of the erythrocyte is 7.5 micrometers. A normal RBC is shaped like a disc with a sunken center. Or we can say they have a biconcave shape, meaning they are basically round, with a center that is depressed in comparison with their edges. This shape helps them to transport gases by increasing the surface area of the cell, allowing more diffusion. Mature erythrocytes have no organelles or no nuclei. This characteristic gives them more room for HB, an important component of RBCs necessary for their function. Lacking nuclei, mature, RBCs cannot synthesize proteins nor divide to form. More cells, erythrocytes contain mostly antioxidant enzymes and structural proteins, where antioxidants, together with other functions, prevent RBCs for oxidation, and the structural proteins allow them to change shape and return to their original shape afterward. This property helps them in better exchange of gases. Look at its name again. Here, erythros means red. So, erythrocytes are commonly known as red blood cells. These cells are named so because of their red appearance. And this red color of the red blood cells is due to the presence of the coloring pigment called hemoglobin. Red blood cells are about 1/3 hemoglobin, a protein that gives them their red color. And hemoglobin is actually responsible for the ability of red blood cells to transport oxygen and carbon dioxide, and most oxygen carried in the blood is bound to hemoglobin. Hemoglobin consists of 4 polypeptide chains of globulin proteins, 2 alpha chains, and 2 beta chains, that each bind a ring-like structure called heme group. And each heme group has an iron atom in its very center. From here, we can get an idea about the fact that 1 hemoglobin molecule can transport 4 molecules of oxygen, since each iron atom combines reversibly with 1 molecule of oxygen. And 1 red blood cell consists of about 250 million hemoglobin molecules, meaning that each of them can contain approximately 1 billion oxygen molecules. All these quantities gives us idea about how efficiently red blood cells play their main role. That is transport of respiratory gases. For diagnosis of various blood-related diseases, RBC count is also very important, where a red blood cell count is the number of red blood cells in a microliter of blood. Normal ranges of RBCs count in adult males are 4.7 million to 6.1 million cells per microliter, and in adult females are 4.2 million to 5.4 million cells per microliter. And we know that these RBCs play a very vital role in our body. Let's look at their functions to understand their importance. Although major function of RBCs is the transport of respiratory gases, it mainly performs 4 main functions, that are transport of oxygen from the lungs to the tissues. Hemoglobin in RBC combines with oxygen at the level of alveoli to form oxyhemoglobin. About 97% of oxygen is transported in blood in the form of oxyhemoglobin. Next is transport of carbon dioxide from the tissues to the lungs. Hemoglobin also has the ability to combine with carbon dioxide directly and thus form carb-hemoglobin. About 30% of carbon dioxide is transported in this form. Also, RBC contain a large amount of the carbonic anhydrase. This enzyme is necessary for the formation of bicarbonate from water and carbon dioxide. So it holds carbon dioxide as bicarbonates. Thus, it helps to transport carbon dioxide in the form of bicarbonate from tissues to lungs. About 63% of carbon dioxide is transported in this form. Third function is its buffering action in blood. Hemoglobin functions as a good buffer. During the conversion of carbon dioxide into bicarbonate, hydrogen ions liberated in the reaction are buffered by hemoglobin, which is reduced by the dissociation of oxygen. This buffering helps maintain normal pH, and thereby plays a role in the maintenance of acid-base balance. RBCs also play a role in blood group determination. As RBCs carry the blood group antigens like A antigen, its presence is responsible for blood group type A. B antigen, responsible for type B, presence of both antigens, determine blood group type AB, while absence of these antigens determine type O, and Rh factor, responsible for positive or negative blood groups. This helps in determination of blood group and enables the prevention of reactions due to incompatible blood transfusion. So this was all about its functions. These red blood cells perform these functions, circulate in the blood for about 120 days before they die, break up, and are ultimately consumed by phagocytic cells in the spleen and the liver. In fact, 2.5 million RBCs are destroyed every second. Although this is only a fraction of the trillions of RBCs in the body must constantly produce new RBCs to maintain homeostasis. The process of producing new erythrocytes called erythropoiesis that we have already explained. To maintain homeostasis, this process of erythropoiesis needs to be regulated, and it is done through a negative feedback loop, where after completing their life cycle, when damaged RBCs removed from circulation, oxygen level falls. Their kidneys detect the declining levels of oxygen and respond by secreting a hormone called erythropoietin, EPO. This EPO stimulates the red bone marrow to begin the process of creating new erythrocytes. As a result, an immature form of an erythrocyte, called a reticulocyte, is released into circulation. After 1 to 2 days, the reticulocyte becomes a mature erythrocyte. And then finally, as the number of RBCs increases, oxygen levels rise. Less EPO is produced and thus RBC production declines. And so the loop continues. And RBCs continue their working for next 120 days. After 120 days in circulation, as an RBC ages, its membrane weakens, becoming fragile. And when it passes through the narrow capillaries in the spleen, it begins to break down. Then the macrophages in the liver and spleen ingest and destroy old RBCs. Now in this process, hemoglobin is broken down into its two components, of globin and heme. Globin is further broken down into amino acids. And these amino acids are then used for energy or to create new proteins. While heme is broken down into iron and bilirubin. Iron is transported to the bone marrow, where it's used to create new hemoglobin, while bilirubin is excreted into the intestines as part of bile. So this was all about erythrocytes, where we discussed from their characteristics till the fate of erythrocytes, which was the first formed element. Now we will move toward the other formed element that are leukocytes or white blood cells and platelets.